Hello everyone, my name is Deja Tillman. Today is April 4th, 2020. This is the lab exercise video for biochemical testing for microbial identification, carbohydrate, fermentation testing. The purpose of this test is another technique to identify unknown microorganisms by the fermentation technique. So pretty much we're going to go over my results for this experiment. I actually did this experiment around, it was exactly 10 o'clock this morning, April 4th. And now it is time to go over the results. So, let me get there are 6 tubes altogether. We had a solution before that was red, kind of like a darker red than this one here. I took glucose powder and added half to each, which I labeled Staphylococcus, epidermidis, and S. cerevisiae. So I added that, shook it around, let it dissolve, and then the next step was to add the micro Durham tubes inside, which you can see here. In order to get the tubes to go to the bottom, you have to screw the cap on and then shake them kind of gently in order to get them to the bottom to remove all of the air. I did 6 of these tubes. I added glucose powder to number 1, fructose powder to number 2, and mannitol to number 3. First, I'll go over quickly the glucose. So the staph epidermidis and the Staphylococcus cerevisiae, they have both fermented with gas production. You can tell by the yellow color that it was successful. So it looks like the cerevisiae is a little bit more brighter yellow than the epidermis. The next 2 tubes, I did the same process, but I added fructose. Here is my fructose added to my Staphylococcus and epidermidis. And as you can see, the tubes are in there, floating to the bottom. They both have fermented with gas production as well. This one has fermented a little bit more than the other. This is the cerevisiae. The last 3 tubes, I added mannitol powder. And we have a little bit different results here. So on the Staphylococcus epidermidis, which is this one here, it fermented a little bit. You can still see the pinkish color, and the tube is also floating to the bottom. The Staphylococcus cerevisiae is close to a non-fermentation. So this is this one right here, because it's very pink, close to red, which is kind of close to the original color I started with. These little mini tubes inside, called the Durham tubes, is what enables the detection of carbon dioxide and gas. Basically, it traps the gas inside. So what I've demonstrated is the technique for identifying unknown microorganism. My fermentation by adding a dry chemical to my tubes, and also adding the pure organisms of the Staphylococcus cerevisiae and Staphylococcus epidermidis. As you can see, bring them in close, and then I will turn them slowly so you can see the colors and the end product. I do want to explain why my last 2 here are different in fluid size is because I did make a mistake and I poured more of the M powder, not the glucose or fructose, but the mucus into my epidermis and cerevisiae. I poured more of the amount of the M powder into this one, so I had to even the powder out and put some in this one. So the results may not be accurate because of that mistake, and I was unable to remove the powder dryly with putting it in the cerevisiae, but these are close results that my assumption would end and there was no fermentation. So once again, my name is Deja Tillman. Today is the same day I started with this procedure, April 4th, and these are my results. As you can see, I'm gonna bring out the pinkest to the yellowest. Thank you guys for watching, and I hope you guys had a wonderful day. Bye-bye.